So today we are going to pick up where we left off with viruses, and I have about 10 minutes reserved at the end of today to talk about next week's midterm. So that's right, we're 1 week away from your midterm. Yay! You know, everyone is just so excited. So we'll talk about that at the end here. All right, so last day we were talking about viral classification, and I had mentioned there's lots of ways to classify viruses. We can talk about their nucleic acids. Are they double-stranded? Are they single-stranded? Are they RNA? Are they DNA? We can talk about their shapes and sizes. Are they icosahedral? Are they helical viruses? Are they enveloped? We can also talk about, you know, who or what they infect. So are they gastrointestinal viruses? Are they insect viruses? And those kind of things. So you can see from this slide, there's an international committee around virus nomenclature, and they're the ones who named SARS-CoV-2, for example. And they didn't necessarily communicate with the people who named the disease. So COVID-19, SARS-CoV-2, a little bit different from one another because these communities of scientists and clinicians were not communicating very well. But you know, that's not unique. If you think about HIV and AIDS, the virus has a different name than the disease, and that's okay. People get used to it. They've got it figured out. So there's maybe about 100 families of viruses that have been characterized, give or take a little bit. I've got an example here. So here's an example of a family. So this is the Retroviruses. These viruses generally infect the gastrointestinal and or the respiratory tracts. And you can see the characteristics around this family: unenveloped, icosahedral capsid, they're double-stranded RNA genomes. So all those things, every virus in this family will have the same. So we can go down to the genus. So in this case, the genus is the ronavirus, and rotaviruses infect the gastrointestinal system. They're pretty common infections in infants and children, and there's a vaccine, and I'm not sure exactly which rotavirus the vaccine is for, but like I said, pretty common kind of infection. Okay, so what we want to do today is talk about, I have 6 viruses that I want to kind of feature specifically. These slides here talk about the families. I care more about the viruses again than the actual families. So if you don't remember that the herpes viridae are part of a certain family, that's fine. Just know which viruses are herpes viruses, for example. And we got Papillomaviruses here as well. So these are the DNA viruses we're going to talk about. You can see there's some other classifications. There's single-stranded DNA viruses. We're not going to talk about those, or these this other group. I'm not even sure how to say that. Papovaviridae. So, like I said, you know, we can't cover every infectious microbe on the planet. So today we're just going to focus on viruses. Here's some of the RNA viruses we're going to look at. So the Rhabdoviridae, that's like rabies virus. The Orthomyxoviridae, that's like the influenza virus. You can see I have coronaviruses on there. And I also have the retroviruses, that's like HIV. So just a quick comment. I see lots of people are arriving late. Somehow Zoom gives me a little beeping noise every time someone arrives to the class. And so please try to join the Zoom meeting on time. It's a little disruptive. I'm not sure how to turn off that beeping symbol or chiming noise. I'll see if I can work on that. It's probably somewhere deep into the settings, and I'll figure it out eventually, but just try to show up on time. That would be wonderful. Okay, let's talk about these virus families. But before I talk about these virus families, I want to talk a little bit about how these different types of genomes work, right? We've got the DNA viruses, we've got the RNA viruses, single-stranded, double-stranded, and all those kind of things, right? So last day, we were talking about these processes, right? We've got the replication, transcription, and translation. So some important processes: if you're a cell, Turns out if you're a virus, you can kind of cheat and get around some of these processes a little bit, really just depending on what your genome is made out of. Somebody is saying here, the meeting isn't loading for many. I had to try 3 different devices. I'm sorry to hear that. I don't know whether that's Wi-Fi on my end or your end. Unfortunately, we're just living right now in the time of where we're really reliant on our technologies, and when it's glitching a little bit, not so much fun. Yeah, so remember this, we've got this, and like I said, viruses, they can do different things. So I'll show you what I mean by that. So it says here, viruses don't have cells, so they can do things a little differently. So this is one of the popular classification systems. It's called the Baltimore classification system. And this is David Baltimore, and he said, hey, why don't we just classify viruses into 7 groups based on the nucleic acid type? So some of these viruses are in group 1, that's double-stranded DNA. Nothing really fancy or new here. It's the same as our double-stranded DNA. The DNA gets converted by transcription to messenger RNA. The messenger RNA gets converted to protein by the ribosome. So no surprises there. So what about group 2? Group 2 is a single-stranded DNA. So what the viruses do is once they get inside the host, they make their single-stranded DNA into double-stranded DNA, and then all the processes basically occur as usual. There's also double-stranded RNA. So you can see in group 3, double-stranded RNA, they skip the DNA step entirely. So the double-stranded RNA is used directly as messenger RNA, and then they're making proteins. Group 4, we have something called the plus single-stranded RNA, and you can see it's getting converted to the negative single-stranded RNA. I haven't talked about what that is yet, so I'll talk about that in a minute. Apologize for that, I meant to talk about it already last day, but we'll get to it in a minute here. But just if you think about your DNA, there's 2 strands of that DNA. You know, when we're talking about molecular biology, often we're talking about the template strand, and then there's the other strand. And so virologists, they call one of those the plus strand, one of them the negative strand. So it kind of depends if your RNA, which strand of DNA it looks like. Try not to get too confused about that or caught up in it. It's not really, I'm not going to ask you a lot of details about this on the midterm. There's also single-stranded, negative sense, single-stranded RNA gets converted to messenger RNA. And you can maybe see where we're going with this. We have these retroviruses. They have RNA that gets converted to DNA, and that gets converted to messenger RNA. And lastly, we have double-stranded DNA. So let me just tell you what the bottom line is here. I don't care if you learn all 7 groups. Go ahead and do that. I'm not going to ask you what all 7 groups are. That wouldn't be very fair. It gets very complicated. This is not a virology class. We have, you know, we're going to talk about viruses a lot. But kind of the key note here is that all viruses, no matter what genome they have, they need to make messenger RNA. So whatever they have, RNA or DNA, whichever strand it is, it gets converted ultimately to messenger RNA, and that messenger RNA will get converted into viral proteins. So it's not a big deal if you don't know all the groups, but I will show you some examples, kind of step by step how this works. What is going on here? Okay, it looks like I was trying to do some labels. Okay, yeah. The groups, my arrows and whatnot moved around, but you can see the groups that these 6 viruses we're going to talk about, which groups they fall into. So we'll get back to these examples in a minute. Oh, there we go. For some reason, okay, it's just animated. So you can see the Papillomavirus and herpes viruses are double-stranded DNA. Coronaviruses are group 4, rabies and influenza are group 5, and HIV is group 6. Okay, so how do DNA viruses work? Like I said, just like what we've learned already, DNA is converted to RNA. We also have double-stranded RNA, single-stranded RNA, and single-stranded RNA. So I'm just going to kind of give some examples of each of these. And I also want to give an example of how a retrovirus works. So I have a slide for each of these. So here's a double-stranded genome. This is kind of like what I showed you before. I gave you a sequence. There's 2 strands of DNA. Notice I have it labeled negative and plus. And you can see if you know your genetic code, or at least if you know one codon, you might know this one here, ATG. So ATG is the start codon. So that is what the plus strand is. It's really just looking for the start codon. So if you see what is happening here, the double-stranded DNA is taken and it's transcribed. This is by an RNA polymerase. And the ATG gets converted into an AUG. And then you can see CGA, that's the same. UCA, so that's TCA is converted to UCA, and TAG is converted to UAG. This goes to a ribosome. The ribosome is going to translate that and make it into a protein. So this is just a very tiny, short example. And then now what do we have? Well, if you take a look at this, we have the DNA can get replicated, and we have viral proteins. You take these things together and you make yourself a new virus. So what about these plus single-stranded RNA genomes? So SS is single-stranded, by the way. So in this case here, we have something that actually looks like the messenger RNA already. So we can skip all the steps. These are the easiest ones. We already have messenger RNA. The genome is in the form of messenger RNA. So it's the same sequence that I gave you before, AUG and so on, and you can see the amino acids that it's producing can go directly to the ribosome. There's one other little step is they do want to copy their genomes, so they make the complementary strand of that single-stranded RNA, and then that gets made back into the plus strand. So again, you know, don't get caught up in all the details here. I'm not going to ask you to reconstruct any of these things here. But like I said, there's that key point that these viruses, they have to do 2 things. They have to make proteins, and they have to somehow copy their genome. And that's going to be done using a combination of host enzymes, and sometimes the viruses have their own enzymes that may do 1 or 2 of the steps. What about the minus single-stranded RNA genomes? So these do not look like the messenger RNA yet. So they have to be converted by transcription to make the messenger RNA. So they have to make the complementary strands. You can see UAC becomes AUG. There's our start codon again for our messenger RNA. And this goes to the ribosome, makes the viral proteins. We just have to finish making our genome. So they have to make the negative strand, makes the positive strand, and then we have more negative strand. And then there's our virus. So like I said, if you're getting a little confused, remember those 2 key points. Remember, point 1 is these viruses, they need to make messenger RNA so they can make proteins, plus they need to copy their genome. So the last one, this is the one that I want you to pay a little bit more attention to because it's a little bit weird, and we're going to be talking about it a lot. This is a retrovirus, and this is what this is how HIV works. And so a retrovirus has some RNA, and it's the plus RNA strand. So you can see there's the start codon right in there, the AUG. But they don't go straight to the ribosome. Something weird happens here, something that we call reverse transcription. So remember, transcription is where we go from DNA to RNA. So reverse transcription is where we're going from RNA to DNA. So this is a kind of a unique thing that happens only through these viruses. So you can see it's making double-stranded DNA from single-stranded RNA. So the virus has its own enzyme that does this. It's called reverse transcriptase, and we will come back to that when we talk about HIV. So the double-stranded DNA, you may know that HIV is a lifelong infection, and that is because now this DNA will actually go and integrate itself into the host cell. So it becomes part of the host genome, and that's why it's a lifelong infection. So at some point, though, the viral genome eventually starts to express itself. It becomes transcribed into RNA. RNA finds a ribosome, and it becomes proteins. So some of that RNA, of course, becomes the virus genome, and then you end up with the virus particles. So just wanted to kind of show you briefly how these things work, and you can see that they're all trying to make viral proteins, and they're all trying to copy their genomes. So remember that key point, messenger RNA to proteins. And you can see I have another little note here, and I said this already. It says this may involve both host and or viral enzymes, so polymerases, RNA polymerases, and DNA polymerases. So a couple other things before we get to our examples. I want to talk about what's going on with these viruses in terms of how they can infect things, right? And you can see in my note here, it says every virus has its own host range. So what does that mean? It means that every virus, some viruses are very, very, very specific, right? So here's an example of a very specific virus, is HIV. So H stands for human. So human immunodeficiency virus. So that means it can only infect human cells, but not just any human cell. It actually has a very specific type of white blood cell. It's called a CD4 cell. So CD4 is a receptor found on certain types of immune cells, and that's all that HIV can infect. So very, very specific. I see there's a comment here. Someone says, what were the 2 points again? Viruses need to make messenger RNA so that they can make proteins. That's key point number 1. Key point number 2 is that they need to replicate their genome. Okay, there are other viruses that are maybe less specific as well out there, but here's another specific virus. This is T for bacteriophage. That infects E. coli. Actually, it doesn't even infect all E. coli, only certain strains of E. coli. Influenza virus A. So this virus here is something we would say is a little bit more promiscuous. That means it is able to bind multiple hosts. So influenza A, this is the one that usually causes the most severe illness in humans, but it can also cause illness in pigs and all sorts of birds. So we all have similar receptors, either in our respiratory systems, or in the case of birds, is actually in the gastrointestinal system. And then you may have heard of rabies virus. Rabies is very, very promiscuous. It seems that if you're a mammal, it can infect you. It doesn't just have to be carnivorous animals. It can actually infect squirrels and all sorts of things. And it's just less likely to get infected by rabies unless you're a carnivorous animal, because it tends to be spread by biting. So I want to show you this virus here. This is a bacteriophage. So I think I had mentioned before that, so this here, bacterio, that means bacteria, and phage means eating. So this really just means eaters of bacteria. So these are viruses that infect bacteria like E. coli. Sometimes people just call them phage. You see that a lot in the scientific literature. And you can see there's a picture of poor E. coli there. And you can see E. coli is infected by, I don't know how many, it looks like probably 30 or 40 bacteriophage. And the reason why I'm mentioning bacteriophage is because a ton of viral research is done in these things for a whole bunch of different reasons. Bacteriophage are very easy to grow in the lab. They're harmless to humans. And so much of what we've learned in virology actually is done in these things. So you see them, they're mentioned all over the place in the textbooks. And sometimes it's not said, but it's implied that the information we learned was actually looking at these kind of infections. So someone has a question saying, can they be used as antibiotics? Sort of. There are some people that experiment around with phage therapy, and I'm going to talk about that a little bit more when we talk about antibiotics in general. So a good question though, but it has been used to treat some infections with success. It's just this story is a lot more complicated than that. There's many, many types out there. I want to show you a few types. I saw some cool pictures out there. This is called the lambdaphage, and that one PM2. That's not a very cool name. MVL2. So again, you've got these geneticists, and they always come up with really boring names and codes and things like that. This is one of my favorite ones, though, the corn dog bacteriophage. I thought those ones were really cute. They kind of look like corn dogs. So, pretty cool picture. So I want to talk a little bit about virus replication cycles. I know we were talking a little bit about their genomes. I'm going to play this video for you here. I don't think there's any volume on it. And you're going to see the basic life cycle here, something called a lytic cycle. So here we go. And you see E. coli down there. And if you watch carefully, you're going to see some of these little bacteriophage. And here it comes. And those little tail fibers, they recognize something on the surface of E. coli, so some sort of lipid. And once they recognize a receptor, you can see in this case here, it's injecting its genome. So it kind of looks a little bit like a lunar lander, but it's a little bit more like a syringe. The instructions go into E. coli, and they say, make more viruses. And you can see that E. coli in the background is kind of exploding with viruses. It doesn't happen this quickly. It's more like a couple of hours for this process to occur. So what you just witnessed there is something called the lytic cycle. So you can see this name here, and I kind of want to talk about 3 types of virus life cycles, and you know, what is going on there. So the word lytic, by the way, it means breaking. So that's what you're seeing. You're seeing E. coli was breaking open at the end there. So you can see the first step is attachment, and by the way, the lytic cycle is not exclusive just to bacteriophage. There are all sorts of human viruses that do the lytic cycle. Anytime there's cell damage from viruses replicating, this is usually the lytic cycle. So the first step, you can see there's some sort of attachment going on. So there's something on the surface of the cell that the virus is recognizing. So if this was HIV, it would be that CD4 receptor. And then once it recognizes the cell, the virus can enter. And in the case of E. coli, remember there's a cell wall. In the case of the bacteriophage, it requires an enzyme to basically get through and inject the genome. So the genome gets in there, and you can see the next step is viral gene expression, which says basically the viral DNA is transcribed. So in the case of bacteriophage, which one was this? Lambda, I think. A number of these bacteriophage are extra nasty, and they'll actually break down the host genome as well. So the entire cell is hijacked and only making viral proteins. So after the virus gene is expressed, you're going to have all those virus parts. So you're going to have the DNA, and you're going to have the viral proteins, the capsid proteins, and any other parts or enzymes that are important. These kind of self-assemble in the cell. And in the case of the bacteriophage, there's an enzyme that will break down the cell wall, and eventually E. coli breaks open. So in E. coli, this takes about 100 minutes or so, and the cell breaks open and dies. So this is how one way that viruses cause disease, right? They're killing your cells. So if you've had cold sores, you know, you've got these blisters, and what you're looking at is little herpes viruses that are literally rupturing epidermal cells and causing the blister. Not all the blister is caused from the virus. Some of the blister is caused from inflammation. So that's your immune system trying to fight it off as well. But anytime there's damage, if you ever have a sore throat when you have a cold or the flu, the sore throat is from those viruses that are they're literally killing cells down part of your respiratory system, and this is from the lytic cycle. So this is not the only life cycle. Oh, there's our little virus. It's free. That oh, I don't know why I have this twice. So apologize. There's a second type of cycle I want to talk about here called the lysogenic cycle. So this is a little bit different, and there's actually quite a few viruses that do this as well, not just the bacteriophage. And lysogenic, I'm not actually 100% sure what the word means. I'll have to look that one up, but I think it means something like sleeping or resting. And what we're looking at is the virus going dormant. So if you see in my diagram here, the very first step is exactly the same. You have an attachment event, so the virus is recognizing something on the surface of the cell, and then the genome was going inside. But in this case here, the genome actually gets integrated into the host genome. So what you're looking at right here, this thing I'm kind of outlining, this is the E. coli chromosome. And you can see over here, there's the host genome and the virus genome becoming one. So the viral genome gets integrated and becomes part of the host cell. So you can see step 3, the host just keeps on living and dividing, and now that virus is permanently a part of that host cell. So this can go on for a while. In the case of many of these viruses, this is actually something that happens with some types of herpesviruses. They do go dormant, but they don't always do it this way. You know, you may have cold sores that go away for a while and then they come back. And my understanding is, I would have to look this up though, but one of the stressors is excessive exposure to UV light. So some people get cold sores in the summer when they get sunburned, and that's the UV light is activating the virus. The virus gets activated. You can see it's kind of the same as my induction step before, where we have the viral genes are going to start to get transcribed. We end up with new virus genomes and virus proteins, and eventually the virus gets released and goes along and maybe infects something else. So somebody's saying, is this what herpes viruses do that? Yes, it's one of the mechanisms that herpes viruses do to go dormant. They do actually have multiple dormant type of mechanisms as well. So someone's asking, is integrated DNA similar to recombinant DNA? Yes and no. Usually when people say recombinant DNA, they will mean something that a scientist has made in a test tube. Because there's different mechanisms around it, but just the terminology, you're right. It's the same idea though. You're taking two different types of DNA and you're attaching them together. But different terminology really means different things. So in this case, this is the virus integrating itself. Recombinant DNA really does mean a scientist is doing it in a test tube. Okay, there's a picture from the textbook showing the lytic cycles and the lysogenic cycle. You can see they're using a slightly different word. I know they do use the word lysogenic cycle in the textbook, but they're saying latency. Oops. So latency kind of means dormancy. And you can see they have the same term there. There's a provirus, and the cells are multiplying, and eventually the new virus particles are made and going to be released from the host cells. Somebody's asking, how does UV trigger a portion of the DNA to activate? Good question. I do not have an answer for that. Probably has a little bit to do with DNA damage. You probably know that too much UV light gives you sunburn, and maybe in the long term it might give you cancer. So UV light does damage DNA. And so I'm guessing with a little bit of DNA damage, the DNA repair machinery gets activated, and somehow that activates the virus. So there's probably a mechanism in there. I'm not fully aware of it, but I'm pretty sure it has something to do with DNA damage. So there's one more life cycle I wanted to mention. This is referring to what's going on with HIV. And this is the retroviral life cycle. So I already showed you this before. I'm just showing you the picture of it happening in a cell. You can see the infection stage, kind of the same. You've got a genome being inserted, but your genome, in this case here, I guess I meant to put a little label on it. I have a little label on it. Thought I did. Maybe I don't. Okay, so this here is my single-stranded RNA right here. And the single-stranded RNA makes double-stranded DNA. So this is my reverse transcription. And you get a provirus, and same idea as before, it can go dormant for a while and then eventually express itself and cause the creation of more viruses. So we'll look at this in more detail when we do come to HIV. I do have a little cartoon to show you though. Here's a virus and here's a retrovirus. So, haha, kind of cute. Thought you might enjoy that. So let's talk a little bit about animal cells and animal viruses, because of course we're animals and we want to know all about those. So what's the difference between an animal cell and a bacterial cell? Animal cells have no cell walls. This actually makes it easier for the viruses to get in because there's no cell wall to penetrate through. We do have a nucleus, and that's important for some viruses. Some viruses don't need to go to the nucleus. Some are fine to replicate in the cytoplasm. Some have to get to the nucleus. So that is a little bit of an extra barrier that may be important for some viruses. Animal viruses, as I mentioned, many more are enveloped, many more than you would see that are infecting bacteria. And there seems to be maybe even a little bit more diversity in animal viruses out there. So I want to talk a little bit about how they get into cells, because there are multiple mechanisms, and I'll show you some little diagrams or animations for each of these. So the first one you can see is direct penetration. So direct penetration, it basically just means the DNA or genome is just getting injected. So if you take a look here, you can see you've got some sort of icosahedral virus. It's binding a receptor and it's just directly injecting its genome. So I'll play this animation. There it goes. And the genome gets inside, and then the genome says, hey, make more viruses. So direct penetration. 2 is something called endocytosis. So what does endocytosis mean? Endocytosis is a process where a cell makes an invagination in its membrane and kind of wraps its membrane around something. So cells will do this. Some cells do this for eating. Our immune cells do this because, to destroy invaders. So what the virus is doing is tricking the cell and saying, hey, I'm some food, why don't you eat me? That's kind of what's going on here. So I'll show you the little animation. So you can see it's binding the receptors. And there's the membrane. It's forming this little invagination, and it completely surrounds the virus. So sometimes this goes all the way to the nucleus. You can see the diagrams on the right. You can see one of them is getting uncoated in the cytoplasm. And you can see the one on the right gets uncoated at the nuclear membrane, and eventually the DNA gets inside the nuclear membrane. So I think my little animation here, maybe it goes another step or two. It gets uncoated, and so it still has the capsid on it. And the pH of the cytoplasm is different than the pH outside the cell, and that actually causes the capsids to basically dissolve. So number 3 is membrane fusion. This may be one of the most common methods. So if you take a look, we've got this little virus. This little virus is enveloped. So remember, an envelope means it has a piece of membrane that it took on its way out while budding away. And so that membrane, the envelope, will fuse with the host plasma membrane. So you can see what's happening there. There it goes, and the capsid gets inside. And like before, the capsid dissolves and the virus genome is released. So somebody has a question here. It says, on the notes, it gives examples of viruses that use these methods. Do we need to remember that? That kind of falls into that gray zone of knowledge that I wouldn't say it's essential for the course, but you never know when things like that might pop up on a test. So usually I'll have a few questions on a test to really reward people that study hard, and that might be a kind of question I might ask as an example of a multiple choice question or two. But in terms of bigger picture, it's not really bigger picture, and I wouldn't worry about it too much. But like I said, there's always going to be a couple of questions like that in the midterm to really reward those that are studying really hard. So how do these things get out? Oh, sorry, I'm just ahead of myself here. So what's going on here? The general rule of thumb is that DNA viruses usually need to enter the nucleus. Why is that? Because think about it, the nucleus is where the DNA enzymes are. So if they want to replicate themselves, the DNA needs to get to the nucleus. Usually, there are exceptions. Again, sometimes viruses have their own enzymes to kind of help them out in the cytoplasm. RNA viruses are usually fine to stay in the cytoplasm. And that's because the RNA enzymes, RNA polymerase, is found in the cytoplasm. Ribosomes are also found in the cytoplasm. And then it says here, I mentioned this before, each will use a combination of host machinery and viral enzymes for the replication. So there's that key point again. All viruses must make their RNA, and the RNA must be read by host ribosomes because viruses do not have ribosomes. So how do they get out of cells? Kind of the opposite of how they get in. So you remember that membrane fusion thing? The opposite of that is something called budding. And like I said, this is one of the most common ways that viruses get in and out, is this membrane fusion, and on the exit, they bud. So you can see there's our little virus capsid. I'm looking at number 1 here. And these little pink things, those are the spike proteins, so the viral glycoproteins. And the virus is departing, it's grabbing some of the membrane, it's becoming an envelope, and then you have the infectious particle there at number 5. Some of them are getting out by lysis. I think we mentioned that one already, and that just means breaking the cell open. You know, so the cell just becomes loaded. Sometimes there's enzymes that will break down the cell wall, or in the case of animal cells, loosen up the membrane, and it's kind of like the cell exploding. So there's a colorized electron micrograph that looks like the influenza virus budding away from the host cell and ready to go infect somebody else. Okay, so before we get into our virus examples, I do have a Kahoot for you. So load up the app and we'll play a little game and see what we know about virus structures. Okay, there it is. Okay, good. I can see some people are joining in. That's great. Give you about another 20 seconds to join in. You can always join in partway through the game. I think I have, this one has 7 or 8 questions. So lots of opportunity to get some points. Okay, here we go. I'm going to get started here. So the first question, can viruses reproduce independently? So the correct answer is no, they need a host cell. They must use the host machinery to replicate. So VT, see if you can hold the lead. Question 2, what are the main components of a virus? So number D, or the green one, is correct. DNA or RNA and a capsid. Some viruses do have an envelope, but that's only some viruses, so we're not going to include that on this here. Okay, the scores are moving around a little bit. What is the capsid around the viral genome made of? So the answer is protein. Oh no, I must have selected the wrong answer on this when I was setting the quiz. Capsid is made of protein, so I apologize for that. I'll have to fix this quiz for next time. Sometimes you copy the questions and you forget to change where the check mark is. So I don't know who guessed phospholipid, you're wrong, but it looks like you're benefiting from getting the points. But the capsid is made out of proteins. The envelope is made out of phospholipids. All right, question 4. Viruses can infect any organism, true or false? So the answer is false. Most organisms do have viruses that infect them, but each in particular virus can only infect usually one organism or sometimes a few. Some are a little bit more promiscuous, such as rabies, but most do have a very specific host range. Scoreboard moving around a little bit. Which cycle is pictured here? Sorry for the typo. Correct. This is the lysogenic cycle. Let's just take a quick look at this picture here. You can see in this particular life cycle, like all life cycles, you got the attachment and the viral genome is getting inside, but then you have that incorporation of the genetic material. So you're making a provirus here. And there's no indication that we have a retrovirus because there's no indication we have any RNA. So this is the lysogenic cycle. Okay, how are those points coming out? Helen in the lead again, and Ariba is coming in hot, it looks like. Question 6, what is an integrated piece of viral DNA called? So the answer is provirus. Sometimes you also see it called a prophage if it's something that's infecting a bacteriophage, but I like the word provirus because it's more inclusive. Scoreboard, getting mixed up a little bit. Question 7, which animal virus has the enzyme reverse transcriptase? So the correct answer is retroviruses. Remember, retro means backwards, kind of like the word reverse. Okay, one more question. What is a well-known retrovirus that negatively impacts humans? So the correct answer is HIV. So well done. Let's see how the scoreboard comes out. Third place, Pam, well done. Second place, Dylan. First place, Helen. So well done, Helen. So I'm seeing Helen's name there a lot. Good job. Maybe everyone else can see what you can do about beating Helen next time. Okay, so well played. Sorry about the typos on that one. I'll see if I can fix that up for next time. Somebody has a question here. Somebody is asking, someone wonders why viruses and it says, why do viruses and whatnot want to make us sick and possibly die? It seems kind of silly because if we die, they die, yeah. So actually, that's a very good question. I'm just going to answer it very quickly, and you can always contact me during my office hours to have a discussion about it. But you'll notice that a lot of viruses that are new to humans, so Ebola, for example, Ebola is not a human virus, not usually. It actually, we believe it's in bats, and the bats barely get sick. Often when they jump species, they tend to be a lot more nasty because well, they haven't had a chance to adapt to our organisms, so they're killing us in a sense almost accidentally. Eventually the viruses will mutate and evolve and often become a lot more mild, you know, until we kind of find you know, it's kind of like, you know, we're compromising with our genomes, they're compromising with their genomes, so it's a little bit of evolution going on is what's happening. So that's kind of the brief, quick, and dirty answer. Okay, so back to our PowerPoint here. Okay, I got to figure out how to share it again. Okay, so I keep wanting to get into the examples, but I keep forgetting about these things that I want to talk about first. Types of viral infections. So we've mentioned some of these words already, and I just want to give you some examples of some of these things. So, you know, in terms of what is going on with disease. So you can see number one is A, and I mentioned that A is the killing of the host cell, and that leads to something called cytopathic effects. So there's a good word for you. It's not going to be on the first midterm, but it'll probably be on the final exam because I'm going to talk about cytopathic effects a little bit more later, or maybe the second midterm. I can't remember where I talk about it a bit more. But this is just mean, you know, basically viruses killing cells. So some viruses, again, like Ebola is a good example where there's cases of people with Ebola that are literally bleeding to death because it's a hemorrhagic virus, which means people are they're vomiting blood and whatnot. And this is the cells dying, right? And then this is very nasty. I mentioned also sore throats from some viruses. That's another example, and it's just the host cell. So this is where we have some of the disease from viruses. Not all the diseases from this. Sometimes it's our own immune system causing things like fevers or aches or chills. And some more on that later when we talk about the immune system. A second category are chronic or persistent viruses. So some viruses, you get the infection and, it runs its course. It's a couple of weeks, it's a week, and, your immune system clears it and you get better, or, worst case scenario, you die. Other viruses kind of, they will infect people chronically. So a good classic example of this are hepatitis B and C. So hepatitis viruses are infecting the liver, and they just have sort of a slow infection and slowly make viruses. And so, they're not killing the person in the short term. Maybe in the long term, the person might develop cancer from this, and that's what might kill them. But the virus is just, it's just growing very slowly. And this is actually a good strategy because now the virus has many, many years to potentially infect other people. Something like herpes viruses, sometimes they will hide. I don't usually use the word chronic or persistent with herpes viruses because they're not causing necessarily any symptoms for a while. They're more dormant, which is our third category. But you can see there's a list of other viruses there that may be causing, chronic symptoms. Sometimes it's basically barely clinical, or whatever, but they can cause long-term chronic infections. Latent is what we're talking about when we're talking about the herpes viruses, meaning that there's an initial infection and then the virus goes into hiding and it's inactive, and it may someday become reactive or it may not. For some people, for example, with cold sores, it's a relatively frequent kind of thing. Some people with cold sores, it's not very often at all. So you've got, again, there's multiple mechanisms going on here. Sometimes it's the immune system kind of holding it at bay. Sometimes there's certain stimuli that are activating the virus and whatnot. But herpes viruses are a good example of latent infections. And then the last one, oncogenic. So you may have heard of oncologists. So there's that word onco, and it's dealing with people that, of course, have cancer. And so there are viruses that, in fact, cause cancer or tumors. So here's an example of one. This is a virus that's infecting a rabbit. So you may or may not have heard of a jackalope. If you don't know what a jackalope is, it's worth it looking up. If you ever go to sometimes if you end up at a hunting lodge, or if you ever are curious, go into a taxidermist shop. And this taxidermists are where people go hunting or fishing and they get their fish mounted, or maybe the head of their deer mounted. And so you see these things, and sometimes if you go into one of these shops, you'll see a jackalope, and this is a rabbit, and it's got little deer horns coming off it. And those are usually just jokes made by the taxidermist. But once in a while, people do find rabbits out in the wild with these things that kind of look like horns. So what are these things? This was investigated in the 1930s, and so this guy here, he said, okay, what is going on with these horns? Because he realized they weren't hard like horns, they're a lot softer. So he ground some up and filtered it and rubbed it on the heads of new rabbits, and the new rabbits got these little horns. And what he realized that these horn things were actually tumors. And so he made the discovery, and again, back in the 1930s, people didn't really know the understanding that we have now of cancers. And so this was a huge kind of event in cancer research where people actually found a cause of tumors. So I'll show you some more animals with horns. And what do we mean by horns? This is the same virus, by the way, that causes warts. And so there is, it looks like a bulldog or something like that. It's got a big wart on its lip. There's an unfortunate cow. And there's a deer, and you can see along its neck here, it's got some big warts. And of course, humans can sometimes end up with warts. Thought I'd find a funny little picture for you. If you want to look up disgusting pictures of warts, you have Google at your beck and call. Go for it. So what are warts? Warts are not caused by toads. Okay, so feel free to handle toads unless you're in Australia. The cane toads have some poison toxins in them. You want to avoid those ones. But most toads are just fine to handle. They're not going to cause you warts. Warts are caused by the papillomaviruses. Kind of a cool name. It sort of runs off your tongue. Pretty much almost everybody will get warts at some point in their life. Sometimes it's very minor and not even, you don't even notice it. Often it's a little bit more common in children and ends up on the hands or the feet. And they're just caused by people that have warts and they shed them. And so kids have more contact with other kids and are maybe not washing their hands as easily. And so the viruses will penetrate the skin and cause these little tumors. So warts, those type of tumors are usually benign, meaning they're not giving you any cancer, but they're causing these growths and they can sometimes be painful. I remember when I was in, I think it was about grade 7, I had one right on the bottom of the arch of my foot. And so that was not very fun, right? Like just basically spending a month walking around and, kind of limping a little bit and feeling this lump on the bottom of my foot. It was actually quite painful, and I didn't do anything about it. I was in grade 7, couldn't be bothered to do anything with it, and actually just went away. Some people have warts for many years. I know one person who had a wart on her, I think it was on the knuckle of her index finger, and it was this white thing. It was very visible, and it was a family member, so I asked her about it, and then she must have had it for 20 years, and then one day she noticed it was gone. So, in that case, it was not really too bad, a minor inconvenience. So I think I have a picture of the papillomavirus here, and somebody's just making some comments about getting warts. Yeah, they're not very fun, but usually, like I said, not too serious. So there are quite a few types of viruses that cause warts. And so we're going to get into our virus examples right now. I'm just taking a look at the time. Don't want to run out of time and finish talking about the midterm. So the first viral family we're going to talk about indeed is the papillomaviruses. And so I'll get right into that. And like I said, we've got 6 viruses I do want to talk about as specific examples. So the papillomavirus, we were just talking about now, there's something like 200 types. Okay, I see on the slide it says about 170 types. So maybe that number needs to be updated. I'm not really sure. They are double-stranded DNA viruses. These particular viruses are not enveloped, so sometimes we use the word naked for that, meaning they don't have an envelope around them, and they enter by direct penetration. So generally, these things are causing warts. Some of them do prefer certain parts of the body. Some of them may cause genital warts. So you can see it says there's 30 to 40 responsible for genital warts. And that's kind of a twofold thing. The genitals, the tissue is a little bit different than other parts of your body. It's a little more tender. And then of course you've got the transmission, right? They can be sexually transmitted, which allows another pathway for these types of warts to get transmitted from person to person. You can see the note there. It says some types may cause cancer. I will get to that in a minute. There are different types of cancers that can be caused by the papillomaviruses. Feel like there's something else I was going to say about them. But yeah, the studies show, if you look at, people's antibodies in their blood, most of the human population will get these kind of infections at some point in their life, a very, very high percentage, whether they're childhood warts or genital warts, very, very high percentage of the population. Even if you didn't know you had them, you may not necessarily know you had the infection. There's the life cycle. You can see, kind of just reiterating exactly what I said, the genome gets in there by direct penetration. This viral genome has to get into the nucleus, and you can see there it is replicating and making the new viruses, and the viruses are eventually released. So you may have heard of papillomavirus. Often it's called human papillomavirus, and it's usually referred to as a sexually transmitted disease. Although, like I said, there are many human papillomaviruses that just cause normal warts as opposed to genital warts. We don't really know the prevalence of this, but there are a few studies that seem to indicate maybe 75, maybe 85% of the population gets these, and by these I mean the sexually transmitted kind. And so that's a huge percentage of the population. And again, some of these warts are spread both ways. It's just human contact or rubbing. Sometimes in the environment, I know that there have been cases, and I think somebody in the comments had mentioned they had got some from the swimming pool. And that's, that's common. People are in the showers and their skin is coming off, and it may contain those virus particles. And if it finds a little crack in your skin, it can get in there and cause some warts. So we're concerned about these ones because you can see the note there. It says some strains cause cancer. So cervical cancer is the big one that we talk about a lot. And the good news is there's actually vaccines available now. So talking to the women in the class, you may know a little bit about getting a Pap smear. And so a Pap smear is basically where a sample is taken of your cervix to look for abnormal cells. So they do a little swabbing, and you can see this is showing on the top a normal cervix and normal cervical cells. And so they do a little swab, they send it to a lab. The lab will stain the cells and put them on a microscope slide, and a technician will look. And if the cells look like this, that's pretty normal, right? And the cancerous cells or precancerous cells, you can see they're smaller, the nuclei are bigger, they're a lot more pleomorphic, meaning they kind of have different shapes and sizes. And so this is a pre-screening for cervical cancer. So if you come back with abnormal Pap smear, you may not be cancerous yet, but it's a good idea to get screened once in a while because it is common enough, and so this is what a Pap smear is all about. So let's talk about the vaccine. The vaccine has been really, really quite incredible. It's been out for, I think, about 12 years now in Canada. And you can see here's some of the options of the vaccines that are in Canada. So you have Cervavix, you've got Gardasil and Gardasil 9, and you can see in the table, for example, Gardasil protects against these strains here. So strain number 6, 11, 16, and 18. So 16 and 18, these are the ones that cause most of the human cancers. So that's why you can see they're in all 3 of these vaccines. And so what they've done is they've added the antigens against, Gardasil 9 is now protecting against 9 strains. And so I believe the Alberta government has changed to Gardasil 9. You can also see that Cervavix was originally only available for girls, whereas Gardasil is girls and boys, and Gardasil 9 is girls and boys. So I usually ask the class, why is this? And, we get a few different answers. The obvious answer is who has a cervix? Well, the girls have a cervix. But the actual answer is that the original vaccine was only tested in girls for that exact reason. It's just cheaper, right? If you think about it, you're doing a vaccine trial, you're looking at thousands of people. And if you have to do girls and boys, then you have to double the amount of people you're doing. So they initially only tested it in girls because they were looking at preventing cervical cancer. Of course, if you do the math, you realize that, sexual activity, of course, does not just involve girls. The boys are involved in that too. Maybe not at age 9, but the idea is we want to give the vaccine before people become sexually active. And the other thing is, it's not just cervical cancer that we are worried about protecting against. And this vaccine has been amazingly successful. People were thinking it would take us 20 years before we get results to see, a reduction in cases of cervical cancer. And we've actually seen there's been a whole bunch of studies done that have shown a reduction in the number of cases, of cervical cancer. So, like I said, here's a list from one study. This is from Alberta Health. And these are the numbers they have. They're getting it from some of the literature. You can see it says it's 99% effective preventing HPV and preventing all sorts of other cancers. So you can see anal cancers, penile cancers, vaginal cancers, head and neck cancers, and a high percentage of genital warts. So huge, huge success for this vaccine. It's really been, like I said, people thought it would take us 20 years before we'd see results, and it's only been about 12 years, and we're seeing really good results in countries that have adopted these vaccines. I'm going to skip that slide. I didn't mean to show it. It was just some more data on what I was talking about. So what is cancer, by the way? I guess I meant to define this a little bit more clearly earlier. Cancer really just means uncontrolled cell division, right? A tumor is a mass of constantly dividing cells. So sometimes a wart can be a tumor. The cells constantly divide, and then they often stop at a certain size, and it may be painful or inconvenient, but not necessarily life-threatening. Cancer is where it becomes life-threatening, where the cells start to divide more, and you know, they may invade other body tissues and things like that. And this has a lot to do with basically just errors in mitosis. And it can be caused by viruses. So what are the causes of cancer out there? I know everyone loves to talk about carcinogens, and usually by carcinogens they mean chemicals. And sometimes people like to talk about UV light, but notice my note here is that somewhere around a fifth to a quarter of human cancers are caused by viruses. So what does that mean? It means that we could prevent a huge number of cancers if we have vaccines. And so the great news is we do have one now for human papillomavirus, at least for some of the strains. There are other cancers that are induced by viruses or related to viruses. You can see I have a Burkitt's lymphoma on this list here, for example. That is caused by the same virus that causes infectious mononucleosis. Most people are not at risk if you're healthy and have a healthy immune system, but if you are malnourished and don't have a healthy immune system, then it is a bit more common. And you can see cervical cancers on this list. So I have cervical cancer bold and underlined because that's the one you need to know, right? If I ask you what cancer is caused by a virus, know the one for cervical cancer. Here is a list of other infectious organisms associated with cancers. We're going to talk about hepatitis B and C, but that's kind of, in fact, it's like the last lecture or the second last lecture, so that's kind of way at the end of the course. But, you know, it's worth it to know that hepatitis viruses do infect the liver, and some of them can cause liver cancer. If people have chronic infections, the cancer eventually happens, kind of thing. Notice HPV, there's a list of some of the cancers it's been associated with. The number one is cervical cancer, but it can cause all those other kinds of cancers because it's usually spread, it's usually sexually transmitted, and so those other parts can be involved in, of course, sexual activity. I do have on this list a couple of other organisms that are not viruses. You can see Helicobacter, involved in stomach cancer, these schistosomes, so these are these flukes. Some of them have been involved in bladder cancers and so on. There are a few other infectious organisms that are associated with cancers, but the relationship maybe hasn't been proven to be so tight. So, for example, sometimes on these lists you see HIV, and HIV isn't necessarily causing the cancer. It's just, you know, it's kind of wrecking the immune system, so the immune system can't fight off the cancers. So is that causing the cancer? Again, that's part of the debate, of course. Okay, so moving on, virus number 2 are the herpes viruses. We talked about these a little bit already last day. I want to just give a bit more detail on that, and just looking at the time here, and then we'll probably talk about the exam after talking about the herpes viruses. So these are also DNA viruses. So notice in this case, they are not naked, they're enveloped. And this is a large family of viruses. I think I showed you the human herpes viruses last day. Let me see, there's the list there. So there are 9 known to infect humans. And I want you to know numbers 1 to 4. I'll come back to the slide in a second. So here's the herpes virus life cycle. It's entering by fusion. Like I said, that's a pretty common strategy. It's getting into the, I think it's getting into the, pretty sure it's getting into the nucleus. It must if it's integrating itself. And I wanted to show you this picture, and this is just kind of for your information only. I'm not going to ask you about this stuff, but I wanted to show you that here's the messenger RNA. And you can see some of the messenger RNAs making these capsid proteins and making the capsid. Other messenger RNA are going to the ER. So we haven't really talked about the ER, that's endoplasmic reticulum, but that's where membrane proteins are made. And you can see here we have our glycoproteins are made in the cell surface. So when this thing buds off, it's coated with these glycoproteins. So here's the herpes virus infections of humans. Like I said, you need to know the first 4. Don't worry about the ones that are not bolded or in red. So let's talk about those. We've talked about our cold sores and our genital herpes. So cold sores and genital herpes are caused by the herpes simplex viruses, number 1 and number 2. Usually cold sores are caused by herpes simplex 1, and usually genital herpes are caused by herpes simplex 2. But maybe about 10% of the cases are swapped around. And again, you know, kind of going back to what's going on with HPV, you know, herpes can be transmitted, you know, from genitals to oral areas by sexual activities as well. So herpes simplex viruses causing blisters. These are lifelong infections. You know, sometimes they're very embarrassing, but a huge percentage of the population does have them. And, you know, they can be kept under control with drugs. In some cases, there are creams and lotions that are for treating herpes viruses. And it's something that, unfortunately, many people have to live with. So number 3 is the varicella zoster virus. Sometimes it's just called varicella, and that's okay. And this is the virus that causes chickenpox. So this used to be a rite of passage. Everybody, it seemed, or at least 95% of the population would get chickenpox, usually by the time you're about 12. Most people much younger, like when you're 4 or 5, kind of when you're, you know, going to playgroup or kindergarten, and you're starting to get exposed to other kids. Very infectious virus. It's spread through aerosol, you know, so it floats around in the air. Very, very easy to catch it. And in this case here, you can see this poor child has a really extreme case of it. They have pock marks all over her body. So chickenpox is called chickenpox because you do get those spots on you. So unfortunately, again, this being a herpes virus, it will lie dormant in your body. And sometimes people get a secondary infection called shingles, kind of it reemerges. And shingles, so chickenpox, most people, you know, in some cases it's dangerous and people have serious complications from it, but that's pretty rare for the most part. You know, you might get a few scars. My one son was unfortunate enough to get chickenpox. He was supposed to get vaccinated for it, and, you know, kind of just before he was about to get his vaccine, he got chickenpox. And he has a scar. If you look very carefully, he's got a scar kind of like right here in his eyebrow. And the hair is not growing right where the scar is. So mostly an inconvenience for him. But like I said, you know, it lies dormant. And sometimes when you get older and your immune system gets worn down, and not even older, like it's more associated with people that are 60 plus, but I know somebody who had it when he was 33 years old and he got sick from something else. And, you know, his immune system got battered down and then he got shingles. So it starts re-emerging, the virus starts growing and replicating, and it does so in your nerves and can give this rash, and it's going to be on the person's chest or back. And in some cases, because it's in the nerve cells, people have an extreme amount of pain. So I know quite a few people who have had shingles, all ranges from ages in their 30s to 50s and their 60s. And some people were, you know, my one colleague had it. He was away from work for 2 weeks, so that wasn't too bad. This guy I know who had it in his 30s, he was away from work for 6 weeks. He had a lot of pain, and it was a quite significant event in his life. So we don't want to get shingles. And again, the good news is, I think I mentioned this before, most children are not getting this now because we have a vaccine. And so now that we have a vaccine, you know, we're not even at the stage where we've had it long enough to know how long the vaccine lasts. We may eventually start to ask people to, you know, get a booster at some point. You know, maybe it lasts 20 years, maybe it lasts 10 years. We're still working on the data for that. And it's basically the same vaccine that you have for chickenpox as you have for shingles. So now when you get to be, I'm not sure what age it is in Alberta, I think it's around 60, they start recommending you get the shingles vaccine, which, like I said, is really just the chickenpox vaccine, probably a little bit of higher dosage because you're a bigger human than when you're a baby, right? Somebody made a comment that she got a booster because she was pregnant and they did blood work and she had no immunity. So probably a good idea. I'm not 100% sure if chickenpox can spread to the fetus, but that's possible. It's just not on my list of things that might do that. So a couple of questions, someone saying, can you get shingles twice? Yes, you can get shingles twice. Not that common, but it does happen. And somebody says, so if you have chickenpox, should you get this shingles vaccine to prevent shingles? Yes, they're recommending it now, like I said, for everybody. I think it's around age 60, it could be 55. And the whole idea is that, you know, eventually you have a good chance of getting it if your immune system kind of gets battered down and you know, it is a long immunity, but eventually it does start to peter away. So I think I have one more slide here on chickenpox. Just showing the infection here, you can see it is a respiratory infection and then eventually replicates in other parts of the body, hides in the nerve ganglia and all that. But I do want to talk about the midterm before I forget. So I'm going to switch to that and maybe I'll come back to this slide next day. So let me find my midterm slides here. Okay, I think we're sharing that now. Okay, so let's talk about the midterm a little bit here. So we're 1 week away from the midterm. And so the midterm is during class time. There will be no lecture during that class time. And it will be found on Moodle, kind of like the quiz was done. I'm going to be using the lockdown browser. And my understanding is that all of you have used the lockdown browser in your anatomy class. So you are at more of an advantage than me because I am still learning how to use it. So what I'm going to do early next week, so in the next couple of days I'm going to have this planned. I'll have it ready for Tuesday for sure, but maybe Monday, is I'm going to have kind of just a test quiz, you know, with a yes or no kind of question. I see some people have saying they have not used it in anatomy yet, the lockdown browser. So I'm going to look into that. Like I said, this is kind of new technology for me. And so if we're having issues, we'll deal with that. Like I said, I'm going to have a test quiz early next week that won't be for any marks. And I want everybody to go into that and just give it a try. It'll have like one question and yeah, just to see how it works. And if the experiment is a huge failure, then we won't worry about it. But I'm told I'm supposed to be using this, so we're gonna give it a try. But like I said, new technology, learning all about it. If it doesn't work for the midterm 1, we'll definitely have it ready and up and running for midterm number 2. So stay tuned for more instructions on that. So what is the midterm going to look like? It's going to be 40 marks. This is usually what my midterms look like when they're 40 marks. They're usually written. So the fill in the blank and short answer, I might tweak those a little bit to be online because I got to think about how these are going to be marked. The multiple choice and the true and false, I know Moodle can mark that for me, but I always like to have some written and a variety of different types of questions because I know some people love multiple choice and some people, it is the bane of their existence. So I want to give you an opportunity to shine in some other parts. Plus the short answer written parts, you know, really give me a good idea of what people are understanding or not understanding in class. And so it's good for me to see the written parts. So I'll talk about that in a a and I have some sample questions sprinkled in here. I will have for everybody, a case study that will be part of the short answer. So by short answer, I mean, what I mean is case study. So it'll be a case study, you know, let's say it's a question and it's giving you some information about a girl with, let's say, chicken pox. And then there will be 5 questions about it. So question number 1 might say, you know, what kind of infection is chicken pox? And you're going to answer, it's a viral infection or something like that, right? So I'll have five questions about it that are related to what we've been talking about in class. So let's talk a little bit about studying. I Googled studying and I came up with this one here, and it's the act of texting, eating, and watching TV with an open textbook nearby. Hopefully that's not it for you. Here are some suggestions about how to study. It is essential in my classes that you review the lecture notes and the lectures. Okay, so I'm saying notes in particular because if you think about my lectures, sometimes I'm giving you information that's not in the notes. So I'm hoping that you've taken some notes on things that I've written down. But other times in the lecture notes, so the PDFs, you know, I may not cover something as much as I meant to, or I'm skipping a few things. So do go through the lecture notes, because this is what I'm looking at when I write the exam. I'm looking at those lecture notes. I'm looking at the study questions at the back. And those study questions aren't usually exam questions. They're usually a bit more open-ended, but I'm looking at those study questions for inspiration when I make the exam. I'm like, okay, this is what was important. This is what we talked about in topic 3, and so on. Make some of your own notes while you study. Don't just read things, okay? Make notes. When you write things down, right, you're organizing things in your brain, and that's helping you to form long-term memories, and that's really important. You want to form good memories so that it's all up here when you need it the most. Make use of resources. There's the textbook, there's the internet. Talk to me about things that you're having a hard time understanding, and hopefully we can work through that. I see we're kind of running out of time here, so I'm going to zip through this quickly, and I'll come back to it next day, and I'll have to finish the virus lecture next day. But I did want to kind of just cover a few things quickly here. So what topics are on the midterm? Topics 1 through 6. So everything on the viruses that we haven't covered will be on the midterm. So whatever we finish covering on topic 6 on Tuesday will be on the midterm as well. So I know I have some slides here for each of the topics. I'm just going to skip through them for now because we're running out of time. I want to show you some sample questions. So here's a sample multiple choice question. So you've seen some multiple choice questions already that I've written. So make sure you do know the language. Make sure you review all the vocabulary words at the end of all my topics. So I'm just going to skip through this here. There's a hard one, by the way. Like I said, I will have a few hard questions in the mix to reward people who have studied really hard. The matching question will be, I'll have 1 or 2 matching questions worth 5 marks each. And usually I have a theme around a question. This one's a bit more open-ended. So the matching question might be all about viruses, for example. Or the matching question might be all about, protist parts. So if you think about all the protist components, we've got the cilia, we've got the cysts. So, usually there's a theme around my matching questions rather than just having all random kind of questions here. Someone says, by lecture notes, you mean extra notes on the bottom of the PowerPoint. By the lecture notes, I mean the PDFs and the PowerPoints. So someone's saying, well, we have the whole class to write the exam or only till 11:30. It's a 1-hour exam. So you have from 10:30 to 11:30. Okay, so there's the answers there. This PowerPoint, these slides, I posted them on Moodle. Fill in the blank. So hopefully that's pretty obvious what it looks like. You can see here all viruses contain a genome made of DNA or RNA and a protein code called a capsid. So hopefully that will be straightforward. Sometimes the fill in the blanks are asking you, give 3 examples of, and so that's another style of fill in the blank. Somebody's asking what will happen if we need more time. Unfortunately, you just get an hour to write the midterm, and Moodle will time out at the end of the hour, and it boots you off is what happens. Okay, so I'm just going to flip through this. So I will go over these slides more on Tuesday. You can take a look at them. They're on Moodle. Just click on exams. The other thing you can look at on Moodle is I have a sample midterm there. So the sample midterm is an old one. but it gives you an idea of the styles of questions and the length of the exam. And of course, online, it's going to be a little bit different and a little bit of a different format, but hopefully the quiz gave you a bit of a flavor of how that's going to work. So somebody says, your sample exam answer and this PowerPoint are different with regards to the DNA in a bacterial cell. I'm not sure which sample answer you're talking about. So anyway, if you have a question about that, please send that to me. I think that's Jody who's asking that, and I'll see if I can correct that. But we are out of time. Sorry for going a couple minutes over. I will see you on Tuesday. Please watch your email. I will be sending out some exam instructions at some point. Hopefully, I will have them sent out before Tuesday so that we can do the sample one on there. I see there's one other question. Somebody is asking about if they qualify for extra time. I can program Moodle on a case-by-case basis if I need to. So that's it for today. I hope you have a great weekend. Please email me any questions you have while you're studying. I will check my email kind of, here and there over the weekend. And hopefully we have some great weather.